Let's study or revise what is photosynthesis. Photosynthesis, we all know, can be carried out by all green photosynthetic organisms which have chlorophyll in them. So, including the autotrophs. So, such organisms are autotrophs. Now, autotrophs means what? The organisms which are capable of producing organic compounds by using light energy and fixing the carbon dioxide. Now, an overview. So, we all know in general what happens in photosynthesis. In the presence of light energy, it produces sugars and oxygen from carbon dioxide and water. So, how does it happen inside the leaf? Why do plants look green? Rest of the light is absorbed and only green is transmitted and reflected. That is why they look green. So, what happens in photosynthesis? You know that it happens in chloroplast in plants. Now, chloroplasts their membranes are rich in a green colored pigment which is called as chlorophyll. Now, they have a number of granular sacs which are called as thylakoids. Now, for light reactions, they happen in the membrane of these thylakoids, these granular structures. So, and the carbon fixation happens at the stroma. The carbon dioxide diffuses in the stroma through the chloroplast and it is transparent to light. Light can easily pass through the chloroplast membrane. So, what happens? The entire process of photosynthesis is divided into two parts. As I said, light reactions and dark reactions. In light reactions, the light energy is captured by the photosystems. So, these are called as reaction centers. So, these reaction centers, they have some few hundred molecules of colored pigments including chlorophyll. So, the reaction centers in these photosystems, photosystem 1 and photosystem 2, which forms the acyclic phosphorylation cycle. Now, in bacteria you will find cyclic phosphorylation. So, I am now going to talk about the light reactions of photosynthesis where the water is broken down to produce oxygen. The photons are captured so as to drive ATP and NADPH synthesis. So, now stroma is the liquid part of the chloroplast and grana are the stacks of the thylakoid. So, this entire light reaction of capturing the photons and then passing on the excited electrons to the electron transport chain, everything is present at the membrane of the thylakoid. So, what happens that as soon as the light is incident on these reaction centers, I will show you a picture. The two reaction centers I said photosystem 1 and photosystem 2. During evolution, photosystem 1 came first and then photosystem 2, but with adaptations to plants to make it more economical or more efficient, the two photosystems both were brought in together. Therefore, photosystem 2 comes first and then the photosystem 1 in that acyclic phosphorylation. So, when the photons are incident, the light energy is incident on the so first comes the photosystem 2 because it was discovered probably later. So, photosystem 2 it is incident, then there are number of pigment molecules. Now, the energy which is absorbed by the pigment molecules, it keeps on getting transferred to neighboring pigment molecules till it reaches the reaction center. What is this reaction center? It is a chlorophyll A molecule which can absorb light at a wavelength of 600 nanometers. So, it absorbs the energy and then this energy drives oxidation reduction reaction, breaking down the water molecule, releasing oxygen and the electrons. Now, this excited electron is then taken up, captured by the neighboring electron acceptor molecule, specific electron acceptor molecule, and then it subsequently keeps transferring the electrons in the electron transport chain which is present at the thylakoid membrane, because of which the hydrogen ion gets released in the thylakoid lumen. So, now once the hydrogen ion concentration in the thylakoid lumen increases, it has to be the electrochemical gradient is there. So, So, this electrochemical gradient through chemiosmosis is then exploited by the ATPase which is present in the thylakoid membrane. Now, this ATPase then generates ATP by driving the hydrogen ions out in the stroma. The hydrogen ions which are then let out in the stroma, they then combine with the cofactor NADP plus forming NADPH. So, this is how the light reactions give rise to ATP and the NADPH. Now, this ATP and the NADPH both are to be utilized for capturing CO2 to convert it into sugars. This we will see how does it happen. So, oxygen, if you see, it is an incidental byproduct because water is available, it captures, breaks down water, and it is an byproduct which happens in photosynthesis. So, if you see the entire picture, whatever I said would be very clear. These are the different, so you must have seen the electron transport even in the chain in your mitochondria. So, it is similar to that. So, the different steps I will repeat. What happens in the thylakoid membrane, the chlorophyll is organized along with other molecules to form the two photosystems. Now, if you will observe that the photosystem 1 it is called as P700, photosystem 2 is P680, which means there is a difference in the maximum absorption wavelength. Now, it is a fact that both the reaction centers are identically same chlorophyll molecules A, but still there is a difference in the wavelength. Why do you think? This difference in absorption properties happens because it is in conjugation with other proteins present in the reaction center. So, because it combines with other proteins in the two photosystems, there is a difference in the absorptive properties. Then the photosystems are what? As I said, these are light harvesting units. So, again to repeat, there are two photosystems. When a photon of light, it will strike the photosystems, the electrons are released. These electrons being high energy states, they are transferred to a nearing electron acceptor molecule, which again transfers it to the electron transport chain present in the membrane, thereby releasing the hydrogen ions in the thylakoid lumen. This causes electrochemical gradient, which is then exploited by ATPase to produce ATP, and the hydrogen ions sent back or diffusing through the membrane into the stroma then are used to reduce the NADP plus to form NADPH. Let us talk about the dark reactions. So, this was the picture which I showed in the beginning. So, we have now spoken about the light reactions. Light reactions have given rise to ATP and NADPH. Now, dark reaction is called as Kelvin cycle. Now, why dark reactions? In this reaction, the CO2 is captured. This CO2 is combined with ribulose 1,5-bisphosphate. Now, in order to combine this, you need an enzyme which is called as ribisco. It is the most abundant organic molecule present on the earth. So, this enzyme then combines the two to give you two stable, one unstable 6 carbon molecule which immediately breaks down to give you two stable molecules of phosphoglyceric acid. Now, because these are the first stable products of the Kelvin cycle, this cycle is also called as C3 cycle. So, in general Kelvin cycle we have three portions. One is carboxylation, then reduction phase, and after the reduction phase, regeneration phase. So, what happens in the carboxylation phase? The carbon dioxide is reduced by ribisco, which is ribulose bisphosphate carboxylase. So, it combines carbon dioxide with ribulose 1,5-bisphosphate to give you two molecules of phosphoglyceric acid. Now, this phosphoglyceric acid is then reduced using ATP and NADPH, and with, if you remember your gluconeogenesis, which is reverse of glycolysis, some of those, it is similar to those reactions. Such reactions takes place to convert your phosphoglyceric acid to 6 molecules of 3-phosphoglycerate. Now, in this then one molecule is then diverted to form your sugars, and the remaining 5 is then again in the regeneration phase. Now, why it is called regeneration phase? Because again the precursor molecule ribulose bisphosphate has to be regenerated. So, now these 5 molecules are again regenerated to give you back ribulose bisphosphate, which can then subsequently again combine with CO2 to give you PGA. So, which enzymes are involved? Ribisco, which is ribisco 1,5 bisphosphate carboxylase. Then it catalyzes the addition of CO2 to ribulose 1,5 bisphosphate, as I said, forming the first stable 2 molecules of phosphoglyceric acid. Then comes the reduction phase. I discussed 3-phosphoglyceric acid is reduced to glyceraldehyde 3-phosphate. If you remember, glyceraldehyde 3-phosphate gives rise to, is a part of glycolysis. So, it is then reduced to give you glyceraldehyde 3-phosphate can then give you sugars, and there ATP and NADPH is needed to reduce PGA to glyceraldehyde 3-phosphate. Enzymes involved are what? 3-phosphoglycerate kinase, these are the major enzymes, and glyceraldehyde 3-phosphate dehydrogenase. Now comes the regeneration phase, where the carbohydrates are fructose and your glucose, and how does it happen? With using enzymes trans aldolase and trans ketolase. If you remember, these are enzymes involved in pentose phosphate pathway. So, again similar reactions of pentose phosphate pathways are involved. Once from glyceraldehyde 3-phosphate you get fructose diphosphate, if you remember your gluconeogenesis cycle of glycolysis. So, now coming back to, we were talking about adaptations. Now, C3 plants, which are generally all the plants follow C3 metabolism. Now, in C3 plants, the light reactions and the dark reactions both are taking place in the same chloroplast, same cell. So, but what happens, as I discussed earlier, that in order to prevent excessive water loss, the stomata is closed. Now, once the stomata is closed, the CO2 now is anyways getting utilized in carbon fixation. So, then a time comes when the relative concentration of CO2 to oxygen reduces. As soon as this happens, ribisco now starts getting more associated with oxygen. Now, what happens? Now, it starts acting as an oxygenase and it will combine your ribulose bisphosphate with oxygen. Now, this oxygen, when it drives this reaction as oxygenase, it will give rise to, I said, a toxic molecule which is called as phosphoglycolate. I will be talking about now this process. Now, once phosphoglycolate is formed, it is a highly toxic molecule. So, the cell drives certain reactions to convert this toxic molecule into a number of molecules like glycine, and then glycine gets converted to serine, and then serine thereby gives rise to many other molecules. Now, all these reactions takes place using organelles, peroxisome and mitochondria. Now, these reactions again happen at the expense of cell's energy. So, ATP is involved and there is a loss of CO2 when the glycine is getting converted to serine in mitochondria. So, effectively 30% of the carbon fixed is lost in salvaging this molecule. So, we will see what do plants do in that case. So, there is one adaptation which is called as C4 metabolism. So, in C4 plants, the photosynthesis takes place in chloroplast, and I was mentioning that there are cells which surround the vascular bundle. These are called as bundle sheath cells. Now, why it is called C4? Yes. So, because the first stable compound formed is malate or aspartic acid. Now, this 4 carbon compound is then transported, now this is slight reaction, is then transported to bundle sheath cells where it again breaks down to release CO2. Now, because it is now very close to vascular bundle and in the inner portions of the leaf of the tissue, it is said to be secured. Now, then once it releases CO2, in the vicinity the relative concentration of CO2 to oxygen is then increased, which then can cause ribisco to again carry out the dark reactions. So this is what is C4 metabolism. Now, so there is a spatial change in the light and the dark reactions. Now then there is one other kind of metabolism which is called as CAM metabolism, which is found in succulent plants. Now why succulent plants? These leaves are plump with lot of water content. Now there is a reason why do they become plump and with lot of water content, because of the 4 molecule which was malate. Now in these CAM metabolism, rather than opening in early hours of the morning and taking in CO2 like C4 plants, and these carry out the photosynthesis and capturing of CO2 to form a 4 carbon molecule at night, and in the morning in the same cells this molecule is broken down and photosynthesis or the dark reactions, fixing of the CO2 which is released happens. So it is a temporal shift between the light and the dark reactions. So what happens in photorespiration? Very hot days, the stomata is closed. Once the stomata is closed, there is no gas exchange. The CO2 is continuously getting utilized. Then again, as I said, relative concentration of CO2 to O2 is reduced. So now rubisco is combining with O2 to give you a toxic molecule, phosphoglycolate. Now this toxic molecules has to be broken down to prevent the cell damage. So now this whole process is called as photorespiration. So as I said, when the CO2 levels drop in the leaf less than 50 ppm, then the rubisco starts acting as an oxygenase. So this is the reactions which take place in photorespiration. So 3 organelles are involved: chloroplast, peroxisomes and mitochondria. If you remember, when I was discussing organelles, I asked you what do peroxisomes, what is the role of peroxisomes? Peroxisomes play a crucial role in photorespiration. So as I said, this glycolate when formed, it is then sent to peroxisomes where the reaction to convert it into glycine happens. Now this happens by utilizing oxygen. So in the peroxisome, the oxygen is then taken in to convert your glycolate to glycine. Now once the glycine is formed, it is then transported to mitochondria and then the glycine is converted to serine. Now this reaction happens by taking in, by release of CO2. So therefore respiration. So as I said, oxygenase and carboxylase is, it depends on their Km value. The Km, although for rubisco to combine with CO2 will be very low or very high. In general, rubisco combines with CO2. So if it can combine with oxygen, the Km value for CO2 will be higher or lower? Lower, more affinity towards CO2 than with oxygen. So what in nature the strategy could be applied to prevent this process, photorespiration? Either the plant should have avoided rubisco, there should have been another enzyme to carry out photosynthesis, or fixing CO2 in an environment shielded from O2. This could be another choice. The third choice could be use of an enzyme that does not react with O2, which is the same. Don't use rubisco, use another enzyme which does not react with O2. So this is what happened. CO2 fixation, we know it happens in the mesophyll cells, palisade cells, spongy cells with air gaps for gas exchange. So now CO2 fixing enzyme is not rubisco. The adaptation happened. Then CO2 fixing enzyme was then changed from rubisco to PEP carboxylase. Then rubisco still remains the same, but CO2 fixing enzyme became, for the first light reactions, it was changed to PEP carboxylase. So we will see how does the malate is formed. So in C4 synthesis, to prevent the wasteful effect of photorespirations, plants like here sugarcane and corn, they adapt to this metabolism. Now what happened, their anatomy, even in if you compare the anatomy between C3 and C4 plants, it is different. In C4 plants, you will see there are bundle sheath cells around the vascular bundle, which is xylem and phloem, while in your C3 plants, and they all mesophyll cells are all palisade. We saw columnar cells and then there were spongy cells. So, the chloroplast in C3 plants and all these cells can carry out light and dark both the reactions. So, now C4 pathway is this, where it combines the PEP carboxylase converts PEP, captures CO2 to make the first 4 carbon molecule, which is called as malate. This malate is then transported to bundle sheath cells in C4 plants, where it is again broken down to release CO2. Now, in this the ribosco is protected. It is not now on the surface so that it can be affected by the oxygen. So, here again then ribosco combines with CO2 in dark reactions to give sugar molecules. Now, what happens in CAM plants? I have already spoken about it. It is temporally different. So, this CO2 fixation into malate happens early hours of the morning, where CO2 is taken in, converted into malate, and then this during the day hours when the stomata is closed, this malate is broken down to give you fixing the CO2, and then CO2 combines with ribosco to give sugars. But then in CAM plants at night photosynthesis, these reactions happen. The CO2 is fixed to give you malate, malate is stored in vacuoles, and then in the morning the malate is then broken down to give you CO2. Now, this happens in succulent plants, I said. Now, why did I say succulent plants? They are more plump with lot of water. There is a large amount of malate present. So, lot of water is sucked in. So, that is the reason why they become succulent in nature. And sometimes there is so much of pressure that the malate is forced to be broken down by such kind of plants.